Hello everyone. I like to welcome in today's session. In previous session, we discussed regarding the variety of terms, explanation of variety of terms, as well as some properties of ideal drug and classification of drug. So, remaining terms, we are going to discuss the remaining terms in today's session. So first term is nothing but the receptor. A receptor, it is a portion of the molecule or structure which the therapeutically active compound interact, producing a series of events leading to an observable response. Means the receptor is nothing but we can say that a portion where actually drug molecule gets attached, and after that there is some reaction and which gives the physiological response. So I am going to show you one video from which idea of the receptor becomes more clear to you. So there are actually the four type of receptors. There are four type of receptors. First type is actually ligand gated receptor. Second one is the enzyme linked receptor. Next one is the G protein coupled receptor. And last one, fourth one is a intracellular receptors. When we considering the ligand gated receptor, this type of receptors especially are responsible for the transfer of the hydrophilic ions inside the cell membrane. So this is an example of the ligand gated receptor. Ligand gets binded to receptor and the receptor gets activated here, and after that it opens its gate and the ions from extracellular fluid or intracellular fluid moves or transfer from one side to another side. So this is a function of this type of receptors. Actually hydrophilic ions, especially Na plus ions, sodium and potassium plus ions, will be passed through such a type of a receptor gate, which is the ligand gate receptor, and model is known as lock and key model. So here sodium ions are moving outside the cell, potassium ions inside the cell. So ligand gets attached to the receptor side, which opens the receptor's gate, and then sodium ion goes inside the cell, which creates the electrolytic gradient. Okay, so sodium ions passes inside the cell, which creates the electrolytic gradient. So this type of function is carried out by ligand gated receptor. Next receptor is enzyme linked receptor, in which actually ligands gets attached to enzyme linked receptor, and here actually the enzyme linked receptor here enzyme linked receptor dimerizes. So example is a tyrosine kinase, which is especially used for phosphorylation. So the ligand gets linked to the tyrosine kinase type of the receptor, and when it get linked to the tyrosine kinase, what's happen exactly there? The dimerization of the receptor takes place. After the dimerization, ATP molecule gets attached to this tyrosine kinase enzyme, and it converts the ATP to ADP for the further reaction. So this is useful for phosphorylation and dephosphorylation. In G protein type of receptor, ligands gets attached to this G protein type of receptor site, and it's having the alpha, beta and gamma type of sub units. So when it gets linked or attached to the G protein receptor, it gets activated and it gets reduced, and the alpha units get detached from G protein receptor, and which is actually the responsible for activation of another protein or enzyme. So this is a function of G protein receptor. And last, or the fourth one type, is a intracellular receptor. So in intracellular receptor, ligands are first passing through the cell membrane, go into the cell. After that, it passes through nuclear membrane, and then it gets attached to the intracellular receptor for the further response. So this is actually 4 types of the receptor. So each and every receptor gives a different type of function, or we can say that they are having the different type of mechanism. And after that, actually the drug molecule gives the corresponding physiological action. So moving towards the next point is nothing but the agonist. What is meant by the agonist? Agonist, it is a chemical or drug that binds to a receptor and activate the receptor to produce a biological response. Means the drug molecule which activates actually the receptor, it considered as a agonist. It's considered as a agonist. Whereas in contrast to agonist, antagonist is nothing but it is again the chemical or drug that binds to receptor and block the biological response. Mean first, agonist is responsible for activation of receptor for biological response, and second creates completely contrast action, which is nothing but it blocks the receptor site and blocks or restrict, or we can say that it stop the corresponding biological response, is nothing but the antagonist. I am again going to show you one video to you. From that, the idea of agonist, what are the types of agonist, how the mechanism of agonist is there and antagonist is there, this idea becomes more clear to you. So I am going to show you that video. So agonist is nothing but an agonist is an drug that binds to the receptor and activates the receptor, which is already discussed here. Agonist is drug which binds to the receptor and activates the receptor, which produce positive response, okay, and which gives the corresponding physiological action. Regarding the agonist and antagonist, both having actually the similar type of activity, but the thing is that the responses are completely contrast. So here actually we are getting exact idea by considering this video. It takes some time to load. So here agonist is coming and gets attached to the receptor. Receptor is in inactive condition. It cannot be links. It's a model actually, and here is actually you can say that the wheel which is correspondingly rotating, not rotating here. When it rotates, you consider its receptor gets activated. So agonist is coming towards the receptor side. When it get attached to the receptor, receptor becomes active and gives the corresponding physiological response or physiological action. So here agonist get attached here and receptor gets activated. But the agonist and receptor interaction is having two type of functions. Means in few cases actually agonist gets attached to the receptor and receptor get activated, but after the removal of agonist the receptor becomes inactive. Receptor becomes inactive means when it is attached to the receptor, then and then only receptor is in an active condition, and when agonist is removed, then what's happened? Then it becomes inactive. So this type of function is also there in most of the cases. In next type of receptor actually agonist is coming, gets attached to the receptor, but the thing is that when it gets attached to the receptor, it creates some covalent changes in a receptor, and then that receptor remains active after the removal of agonist also. Here actually we see the receptor is coming slowly, receptor is coming towards the agonist is coming toward the receptor, and then the receptor gets activated, but after the leaving the agonist, what's happened? Receptor is become inactive. So this one is the first case. Okay, this one is the first case. In second case, agonist is attached to the receptor and receptor is active, receptor is still active, and agonist is removed, still the receptor is active. So this happens due to the covalent change made by agonist in the receptor side. So after the removal of agonist, the receptor remains active in second type of reaction or second type of phenomenon or mechanism. So with this agonist, we are moving towards the actually antagonist, which gives the completely contrast type of activity. Okay, contrast type of activity. So we are moving towards that agonist. So here actually we are getting the idea of the agonist receptor interaction. Slowly the video is moving, that's why it takes time. So we are moving towards the interaction of the receptor and antagonist. Due to the something, I am playing the video once again. But due to some problem is not working. Some disturbances are here in the video. So you can see the previous video, agonist is removed and still the receptor is working. Next, what we are talking about is nothing but the antagonist, which gives the complete contrast type of the action. Complete contrast type of the action. So the agonist is nothing but when it binds to the receptor, it prevents the activation of the receptor. Okay. So here we are going to see what are the types of the actually antagonist and how the antagonist is working here. So you can see that the video is slowly moving, that's why the next picture is not coming due to some connectivity problem maybe. So here the receptor is here and antagonist is coming, which is nothing but the competitive antagonist. When it comes, it blocks the receptor side. It blocks the receptor side. So if the agonist is coming, there is no availability of attaching site for the agonist. So agonist is coming, but it can't attach to the receptor, and the receptor can't get activated. It remains as it is. This type of antagonist is called as or known as the competitive antagonist. Why competitive antagonist? Because it acquires or it accommodates the place where actually agonist gets attached. ज्या ठिकाणी अगोनिस्ट अटॅच होणार आहे ती प्लेस आहे तो अगोदरच येऊन काय करतो, अँटागोनिस्ट आहे तो अक्वायर करून ठेवतो. त्याच्यामुळे काय होतं की जर का अगोनिस्ट आला तर अगोनिस्टला बाइंड होण्यासाठी किंवा आपण म्हणू अकोमोडेट होण्यासाठी देर इज अ नो प्लेस अवेलेबल. सो दिस ऍक्शन or this type of phenomenon is given by the competitive antagonist. We are moving towards next antagonist, which is nothing but the non-competitive antagonist. But before that, you just read the competitive antagonist. What is the description of competitive antagonist? Okay. A competitive antagonist is any pharmacologic antagonist that competes with the binding of agonist at the binding site. Okay. So slowly we are moving towards the non-competitive antagonist. So non-competitive antagonist is any pharmacologic antagonist that binds to the site on the receptor other than the agonist binding site. Means it cannot bind to the receptor side of agonist, but it attached to the different site of the receptor, and the side of the agonist or the attachment site of agonist is remains as it is. So the agonist come and will attach to that particular site. But what's happened, we will see in few seconds. What's happened actually after the attachment of non-competitive agonist? Agonist is coming here and attached to a different site, okay, and the site which is available for the agonist is as it is, okay. So agonist is coming from the vicinity. Agonist is coming from the vicinity and it will get attached to the receptor side. But the thing is that it deactivates the agonist. Means antagonist which is the previously attached to receptor deactivates agonist and receptor remains in unactivated condition. So from this figure actually we are getting exact idea, from this picture or from this video we are getting exact idea how the non-competitive antagonist and agonists reacts with the receptor, and still the receptor is remaining inactive. Next one is a chemical antagonist. Chemical antagonist is a different type of antagonist. It cannot react with the receptor, especially it reacts itself with the agonist. Means antagonist reacts with or binds with the agonist, and as it binds to agonist, it changes the shapes of the agonist. It changes the shape or geometry of the agonist, and then that particular agonist becomes unable to attach to the receptor side. So in previous two conditions or two mechanisms, we consider attachment of the antagonist to the receptor side in a different way. But here what's happened? Agonist it grabbed by antagonist, or you can say that antagonist it gets attached to the agonist, okay, and which blocks the, or we can say that which alters the geometry of the agonist, and then the agonist becomes unable to react with the corresponding receptor side. Slowly we are moving, again video is moving slowly, so that's why exact picture is not in front of you. Okay, so here what's happened exactly? the chemical antagonist reacts with the agonist, it blocks its groups or alters the geometry, and then it is not possible that there will be possibility of attachment of agonist to the receptor side. So this type of function or this type of activity is carried out by chemical antagonist. Okay, so you can see here the restriction of this. And next one is a physiological antagonist. The fourth type is a physiological antagonist. Any drug or the chemical that has an opposite effect. Any drug or chemical that has an opposite effect but through completely different physiological pathway, then we considering that type of the antagonist as a physiological antagonist. So I think from this explanation the idea regarding the agonist and antagonist is become clear. Okay, so we are moving towards the next slide or next point, which is nothing but the drug potency. So drug potency is nothing but it can be defined as the maximum biological action which can be caused by a minimum amount of drug administered. So from this definition I think idea becomes not that much clear. So I will give you one example. Suppose I am considering drug A, drug B and drug C. Three different drugs are there. Drug A, drug B and drug C. Okay. All these three drugs are showing analgesic action, means pain killing action. Okay. But for the pain killing action, drug A or the amount of drug A required is a 500 milligram. For the same action, B drug required, amount of B drug required is a 200 milligram. And for the same action, amount of drug required C is 100 milligram. Okay, so for the same action what's happened here, we require 500 milligram, 300 milligram and 100 milligram. So we can say that if the drug gives the same action, the drug C is more potent than drug B and drug B is more potent than drug A. So when we compare these three type of things, then this idea becomes more clear. Means minimum amount of drug, it gives the desired effect or maximum biological response, then we considering that particular drug is a potent drug, and which is nothing but the drug potency. Okay, then we considering the bioavailability. Actually what's happened, we are taking many number of drugs by oral route. What is meant by oral route? Means by mouth. Okay, so when the any drug is taken by oral route, means by mouth, what's happened? In our gastrointestinal tract, means in a stomach and other parts, okay, of gastrointestinal tract, variety of reaction takes place. Mostly the hydrolysis, oxidation, reduction and esterification, so on. So this type of reaction takes place there. And along with that, what's happened? Drug get absorbed. So when drug get absorbed, it goes into the bloodstream. It goes into the bloodstream and other body fluids. So what's happened? Suppose I am taking 500 milligram of the drug by oral route. Actually, in blood or in a body fluid, there is availability of drug is not a 500 milligram. Is less than 500 milligram. Is less than 500 milligram. Suppose you can say that 300 milligrams drug gets actually distributed in a body fluid. So if we require actually 500 milligram of the drug, if we require 500 milligram of the drug exactly in the bloodstream, then what we do? We use the another route of drug administration, which is nothing but the intravenous route. Means what? Actually, drug is administered into bloodstream by the injection. And in that case, we are giving the dose of the 500 milligram. So that much dose becomes available in the bloodstream. Okay. So this is related with the bioavailability, because the response of the drug is related with the bioavailability of the drug. Though we are taking specific dose, means 500, 300, 100 milligram and so on, but actually bioavailability of drug is different for each and every person. So when actually we are taking it by the oral route, म्हणजे आपण जेव्हा ओरल रूटने ड्रग घेतो तेव्हा वेगवेगळ्या माणसांमध्ये वेगवेगळ्या पद्धतीने ऍब्सॉर्प्शन होतं आणि त्याच्यामुळे त्याची बायोअवेलेबिलिटी आहे ती वेगवेगळ्या पर्सनला डिफरंट असू शकते. टू अवॉइड दिस थिंग डॉक्टर काय करतो, डॉक्टर गिव्स द इंजेक्शन बाय इंट्राव्हेनस रूट डायरेक्टली ब्लड स्ट्रीम मध्ये. ओके, नेक्स्ट बघूया आपण ड्रग टॉक्सिसिटी. Drug toxicity is related with the toxic nature of the drug. So if the drug is given, if it creates some toxic effect, means what? It damages the tissues, it damages the cell, it damages the physiological process. So when this type of damage takes place, it cannot be altered, it cannot be reversible. So when the tissue, cell or the process get damaged, it cannot be recoverable, it cannot be reversible. Okay, so this type of action is nothing but the toxic action or drug toxicity. Okay, next one is the drug addiction. What is mean by the drug addiction? Because we are taking many number of drugs, and most of drugs are related as a or classified as CNS drug, means central nervous system drug. Previously we consider it's in a pharmacodynamic agent, and when we considering the CNS drug, central nervous system drugs, drug actually available shows three type of action, especially for the CNS drug. One is it stimulates the central nervous system, it depresses the central nervous system, then it modifies the central nervous system. But this drug actually may creates the additive type of the response, may gives the additive addiction, it gives the addiction. Okay, so the addiction giving or the drug which gives or drug becomes, or आपण म्हणतो की एखादा माणूस ड्रग ऍडिक्ट झालेला आहे, मीन्स व्हॉट, की त्याला त्याची सवय झालेली आहे आणि इफ that particular drug is not given to that person, it becomes it comes into trouble. So this type of drug or this or the phenomenon is considered as a drug addiction. So many number of CNS drug comes under the category of the narcotics, and in a strip of that particular drug there is a one notation is NRX. So if we demand that particular drug into medical store without the prescription, medical person cannot use the drug. Okay, so unless and until you have the prescription, there is no actually permission to medical store to sold that particular NRX. Okay, so this is regarding the drug addiction. Okay, next is a term is nothing but the spurious drug. Spurious drug. When we considering drug is a spurious one, first condition, if it is imported under the name which belongs to another drug, means फसवलेला असेल, means the name is different and drug is different, then we can consider it as a spurious drug. Suppose if it having the imitation, means डेकोरेटिव्ह काहीतरी केलेलं असेल ड्रगच्या टॅबलेटला, सिरपला, बॉटलला, ओके, अँड त्यामध्ये असलेलं ड्रग आहे इज अ सबस्टिट्यूट ड्रग, म्हणजे ते ड्रग जे आवश्यक आहे ते ड्रग नाही तर दुसरेच ड्रग त्याच्यामध्ये असेल, तरी सुद्धा आपण म्हणू शकतो द ड्रग इज अ स्पुरियस ड्रग. ओके, अँड इफ द ड्रग लेबल जे ड्रग वरती असेल ते ज्या कंपनीचं असेल ती कंपनी अस्तित्वातच नसेल, such a type company is not existing, then we considering that drug is also spurious drug. Okay, and next is there is a label which company is existing, but company cannot produce, that existing company cannot produce this drug. म्हणजे फसवणूक ज्याच्यामध्ये केली जाते हे सगळे जे ड्रग्ज आहेत that drugs are considered as a spurious drug. Okay, next is a term is a misbranded drug. So regarding the misbranded drug, first terminology is if it consists filthy, putrid or decomposed substances. Means a tablet strip, on the tablet strip there is a written as 100 milligram of ibuprofen. But the tablet consists only 10 milligram of ibuprofen. But the size of tablet is more due to the some other material which is a filthy material. Okay, then we considering this drug as a misbranded drug. म्हणजे साईज वाढवलेली आहे पण कंटेंट ज्याच्यामध्ये कमी आहे, ऍक्च्युअली जे त्यांनी सांगितलेलं आहे तेवढं कंटेंट नसेल तर वी कन्सिडरिंग इट्स एज अ misbranded drug. Next one is a drug if it is prepared or packed or stored in unsanitary condition. Means what? सॅनिटायझेशन न केलेल्या कंडिशन्स मध्ये कुठेतरी त्याचं ड्रगचं प्रिपरेशन होतंय, तयार केलं जाते, तिथेच पॅकिंग केलं जाते, तिथे स्टोअर केलं जाते. आणि नाउ डेज वी आर टू मच अवेअर अबाउट द सॅनिटायझेशन. ओके, सो वी आर युझिंग द सॅनिटायझर अँड यू नो दॅट द application of the sanitizer in this actually pandemic situation. What is the importance of the sanitization? Okay, next condition is a container is composed in any poisonous or harmful substances. म्हणजे जर कंटेनर आहे त्या कंटेनर मधले जे सबस्टन्सेस आहेत ते सबस्टन्सेस जर का पॉइझनस असतील आणि त्याच्यातूनच हे ड्रग येत असेल तर मे बी देयर इज अ डिकम्पोजिशन, देयर इज अ वी कॅन से दॅट समवॉट इम्प्युरिटीज और पॉइझनस थिंग कम्स इन टू द कॉन्टॅक्ट विथ ड्रग and in that case also the drug is considered as a misbranded drug. Okay, next one is if it bears the color which is not approved. In we are using many number of color, food color, okay, but for drug only some colors are approved by the authorities. Okay, why? Because drug dye or the color is also chemical compound. So it may react with the drug molecule, it can change their structure, it may having some reaction with this particular drug and drug gets damaged. Okay, so that there are some only few colors are there which are approved by authority, and if the drug having different color which is not approved by authority, then also we considering as a it as a misbranded drug. Okay, then we are going towards the Pharmacophobia. We are familiar with actually encyclopedia. In encyclopedia there is a name or there is a term, so you get any type of information related with the term in that encyclopedia. So pharmacophobia is a similar thing. Pharmacophobia is only restricted to the drug molecule. So you are getting the idea of preparation, what are the effect, what are the side effect. Okay, so it is one authentic book. This is one authentic book where such a type of information is published. Okay, and various authorities publishes such a type of books are considered as a or known as a pharmacophobia. So it is a book of directions and requirement for the preparation of medicine. It is generally published by an authority. Okay, so first one is a British pharmacophobia. First one is a British pharmacophobia. It is considered as British pharmacophobia was the pioneer in this field, and it is published in 1883. It is published in 1883, and since then till today actually British pharmacophobia is considered or rated as the most standard pharmacophobia. You can see the image of British pharmacophobia. Okay, next pharmacophobia is a United States pharmacophobia. Okay, it is published actually in 1920. It is published in 1920 and it is executive and it covers all pharmaceutical chemicals, raw materials as well as drug formulation, cosmetics, vitamins and hormones. You can see the image of the US pharmacophobia. And next we considering the Indian pharmacophobia. Okay, so Indian pharmacophobia is published in 55, and it has having the large number of crude drugs and their preparations. Okay, so you can see the image of Indian Pharmacophobia. So this is a 2018's Pharmacophobia. After that date, depends upon the updates, the Pharmacophobias are updated. So with this a few 5 points, I am going to conclude this lecture. Thank you for your patience listening. Just I am going to recapitulate the points which is discussed during this session. First, we discussed the receptors, 4 types of receptors. Then we consider the agonist, what is the action of agonist, what is the action of antagonist, and then we consider the drug potency as well as other terms related drug toxicity and drug addition, as well as the book which is the authentic book of the drug, which is nothing but the Pharmacophobia. Okay, so thank you all of you. Thank you.